Can you guys see my screen?
Yes, ma'am.
Am I okay? So in today's class, we will continue with the topic of biomaterials, right? And in the previous class, we talked about the bulk properties and also started with what are surface properties people study for these biomaterials. So we'll continue with the surface properties topic. So under surface property, like depending on what type of surface the biomaterial has, we can like make a separate class of biomaterials as non-fouling like materials, non-fouling. Non-fouling means those surfaces on which there is no addition of cell or protein is possible. Means these materials have a bio-inert characteristic. They are completely resistant to any kind of cell interaction or protein adsorption. So you can use these materials for masking your, say, any biological component like protein or anything like that from the immune system. Because if immune system's antibodies and all won't be able to adhere to it and like recognize your biological material in question, either cell or new kind of protein, etc. Now, in general, what we observe that we have already seen that hydrophobic surfaces they bind cells very strongly. Okay? And if the cell is able to bind, the protein is first adsorbed and on top of which cells bind. So it is only possible for hydrophobic surfaces. Whereas for hydrophilic surfaces, it is seen that uh, protein cannot adsorb properly, and if protein cannot adsorb, then the cells cannot adsorb. So these two statements is true for like some degree of hydrophilicity and hydrophobicity. Okay, we studied one thing like ended our lecture, the previous lecture, with one note that we need to optimize the level of hydrophobicity and hydrophilicity to optimize how much cells will adhere on the surface. Okay? We need both of these characteristics, but at an optimum level. If it is very much extreme, the if hydrophobicity is very much extreme at the highest level, then the cells won't adhere. Also, if the surface is very, very hydrophilic, then also cells won't adhere. So in this lecture, we will understand the mechanism of why we see this kind of observation when we do when we use this kind of materials. So non-fouling surfaces, it offers the possibility of inhibiting the formation of conditioning film and thus bacterial adhesion. So just like our mammalian cells, they need a protein layer adsorbed first and top of which the cells adhere. Similarly, the bacteria also interacts with the surface via an adsorbed layer of protein. So first, bacteria secretes some protein. This protein adsorbs to the surface, okay, and it forms something called a conditioning film. Once this conditioning film is formed, the bacterial cells will stick to it. And once it sticks, it spreads, it will start forming the biofilm. And once the biofilm is formed on any surface, then you cannot remove it very easily using, say, antibiotics. So, the formation of biofilm can be very, very dangerous for those kind of medical devices that are going to be implanted inside your body for a long period of time because it can like cause septic condition in the patient. So we don't want when we don't want this kind of biofilm formation on a biomaterial surface, we tend to use a what are we modify the surface in such a way that it becomes non-fouling, okay, or resistant to cell or protein adhesion. The common approach to make the surface non-fouling is to make the surface highly hydrophilic. Okay, note we don't make the surface very hydrophobic, rather we make the surface very, very hydrophilic. Okay. Now let's understand what these 3 points is saying, because these 3 points is going to help you uh, understand why this observation is prevalent in nature. So hydrophobic surfaces have a very strong tendency to adsorb proteins irreversibly. So driving force for the protein adsorption is unfolding of the protein on the surface, accompanied with the release of many hydrophobically structured water molecules from the surface, leading to a large gain in entropy for the system. So what happens is on the surface, some water molecules they are hydrophobically attached on the surface, and their hydrophobic interaction is happening. Now, these molecules are under like very weak bond. Hydrophobic interaction is a weak bond, but when the protein in the solution comes close to the surface, these water molecules are very happy to leave. Okay, so once they leave, then the protein they come close to the surface, it quickly unfolds and adheres and adsorbs, okay, very strongly, and it cannot then you know go back to the solution. The adsorption it has happened irreversibly. So due to release of the water molecules from the surface, there's a net gain in the entropy for the system. If you understanding what is entropy, then is good because it's coming from thermodynamics. Then, according to the second law of thermodynamics, uh, the whatever reaction it happens spontaneous. Spontaneous reaction is uh, supported. So here also, this gain in entropy is basically supporting the adsorption of more and more protein molecules. Okay, system is always trying to increase its entropy, and in this case, for hydrophobic surfaces, the water molecules are very much happy to leave the surface. So the protein in the solution they can adhere, and the adhesion happens in such a way that it unfolds completely. Okay, so very, very strong adhesion is happening here. Whereas in hydrophilic surfaces, so surface hydrophilic, so there are groups, chemical functional groups, which contains like oxygen, nitrogen, etc., that can easily interact with the water molecules in the solvent. And here is hydrogen bond is formed. Hydrogen bond is much more stronger, right? So they do not, these water molecules, they do not have like the feeling that they are not very much easy to detach because they are loving that area. They are strongly bound to that surface. So because of which there cannot be a net gain in entropy. So there is no net gain. So the driving force for the protein addition is basically negligible. All right. So the protein in the solution, like in serum, etc., if even if it comes close to the hydrophilic surface, it cannot adhere. All right. So similarly, if people have observed that on a surface where the protein has adhered, it always adheres as a monolayer, meaning it cannot, uh, one layer of protein molecules that has adhered, so on top of it, another layer of protein molecules cannot form. Okay, so they do not adsorb non-specifically onto their own monolayers. So why this is happening? Because once the protein layer is adsorbed, so the hydrophilic groups in the adsorbed protein molecules, they have a strong tendency to capture the water molecules strongly, bind to the water molecules. So here, these water molecules don't want to leave. So because of which the solution proteins, they cannot again come back and adhere on it. So the net gain in entropy is negligible, like negative, it's towards the negative side. So this kind of reaction is not thermodynamically favorable. Okay. So one way of making any surface as non-fouling is to coat it with a material called polyethylene glycol, or also known as polyethylene oxide. Because if you see the structure of this particular polymer, you will see a lot of alcohol groups, OH groups. So these groups are water-loving groups. Okay, so if you coat it, then the surface will become hydrophilic. This is a very common way of making a surface non-fouling. So what people have observed, if a surface is coated physically or chemically, okay, so you can either do chemical adsorption of this polymer, which is reversible, means you reversibly coat any surface with PEG or polyethylene glycol, or you chemically immobilize this polymer onto the surface. Then what people have observed is it makes the surface very good in order of it very it repels it has a it gains a very good tendency to repel the protein from absorbing, and this kind of behavior is observed only for those PEG polymers which are having a molecular weight of 2000 or more than 2000. So higher molecular weight polymer has to be used to make a surface non-falling. So what driving force comes into the picture? First is since these are polymers, no? So these are chains. So these polymer chains, like they do not want to compress under. So if the protein tries to adsorb on it, they will try to compress the polymer chain. So this polymer chain gives a much greater force to resist the protein adsorption in the opposite direction. Okay. Also, these PEG molecules, they strongly bind water molecules. Okay. So if the water molecules are not released, then also the protein adsorption won't happen. All right. So for making any surface non-fouling, you can simply use PEG, okay, or polyethylene glycol to coat it. Or what people do like for example, if you want to transport uh, beta islet cells for insulin uh, secretion for local secretion of insulin at a target site. So and you want those cells to be hidden from the immune system. So you can make capsules out of PEG, okay, polyethylene glycol. So in this way, the capsules they will not be recognized by the immune system because no protein is getting absorbed on it. Okay, and also if the immune system is not recognizing it, then the circulation time of the capsules will increase. And PEG is also non-toxic and cytocompatible. Okay, so it does not create and it is biodegradable as well. So, so it is good for tissue engineering applications or for drug delivery applications. So in this way, you can use PEG, and you can look for other examples as well, which are used for making the surface non-fouling. Now, another, when you're talking about surface properties, then we should also look into the topology. So, topology means how the surface looks like, whether it is rough or it's smooth, that if there is a texture, like people give patterns, zigzag mesh patterns. So that is the topology, how the surface looks like in simple words. So that thing can also influence how the cells is going to react to that particular biomaterial. Now, the topological features means surface means morphological, you can say morphological features, how the surface looks like. It can vary in length scale. Means your surface topology can be in the length scale of around less than 10 micron. Means whatever roughness is there, the arcs, you can say, you can imagine it as peaks, small peaks or small valleys. They can be, their sizes can be as small as the size of one cell. 10 micron is the size of one cell, or it can be higher than that. That is 10 to 100 microns. Means cells cannot really means so cells cannot sense it individually, but when cluster of cells is there, then they can sense that change in topology, or it can be large topological features like greater than 100 micron. So um, I planned to discuss one paper with you guys where they were changing the surface topology by using techniques like nano imprinting, etc., to modulate the cell migration. Okay, but I don't think I can because there's not much time left for the course to finish. So, but this statement, no, what is this statement is saying? Similarly, it is hinting to those kind of work where people change the surface so they can make nanoscale, say, concentric circles, or they can change the add irregularities to the surface or make it entirely smooth and see and allow the cells to grow. So they will see whether the cells are proliferating or differentiating or migrating. Okay? So these all things can be changed without any addition of bioactive factors like growth factors, etc. Okay. So we can add it. So the example that I gave, like nano imprinting, etc., they these techniques they directly influence the cellular fate processes. But you can also use techniques that indirectly change the cellular fate processes by changing the surface topology. So one such example is called molecular imprinting, okay, where we modify the surface in such a way that it absorbs specific proteins only. Okay, so let me know if I'm going too fast. Okay, because then it will help me only to slow down and come up to the level of your understanding. And if you have any queries, then feel free to ask at any time. So, what is this technique? What is this molecular imprinting technique? So, what people do is, so this diagram here will help you understand what is molecular imprinting. So, suppose you want, this is your target protein molecule, okay, and these are some polymer monomers, and this target protein molecule you have like allowed allowed these monomers to bind to this protein molecule at particular sites. Okay, and in presence of this structure, you cause polymerization of your polymer. Okay, then what will happen when you do extraction of this target protein molecule? You remove this protein molecule, then the sites where these specific groups have bound, those groups they are permanently attached to their protein molecule. Now, this target protein molecule, you were like engineering your polymer. So, if this protein molecule is found in your, say, any biological fluid or etc., then it will this particular polymer will preferentially go and bind because you have already created specific sites where the conjugate sites means complementary sites are available and it can bind it reversibly. Okay, so let me repeat what is happening. So this is your target molecule, any protein molecule, and it has some binding sites. Okay, and to these binding sites, you engineer some chemical molecules, which can easily bind to these binding sites. Okay. Now, what you do, you want to add these chemical molecules onto your polymer, so that in your polymer the specific type of chemical molecules are added. Okay, and you polymerize your polymer in that way only. So you're creating sites, you're creating binding sites in your polymer, if you imagine in that way. Okay. Now, when you use that polymer for say any application, okay, and eh, and where in that application you want only a specific type of protein to adhere, and if that doesn't happen, then your specific type of cells won't adhere. So if you're very specific about that, then you should use this technique. So say you want to make sure that only the protein called laminin only adheres to it. So you have to first figure out the bindings, what are the binding sites possible in the laminin, and what are the functional groups, chemical groups that can bind to the binding sites of the laminin. So you add those functional groups in your polymer, and you synthesize the polymer in such a way that those functional groups are added. So whenever laminin is present in your solution, only that will go and bind to your polymer surface. Okay. This is called molecular imprinting. I hope it's clear to you guys. So here you are indirectly going to modulate the cellular fate process through changing what kind of, or through controlling what kind of proteins are getting adsorbed on the surface. So the previous thing that we discussed, where hydrophilicity and hydrophobicity was coming into picture, to like control the protein adsorption. So there the protein adsorption was happening non-specifically. Okay. So any type of protein can adhere, and if there are multiple types of protein, then there will be some competition going on for the space on the surface. But here molecular imprinting is allowing you to specifically like control which type of protein will adhere on the surface, and then will help you control the cell adhesion on that surface. Okay, so there are various other ways also apart from molecular imprinting, which will help you to change the surface properties. Okay, so one way is crafting your polymer with a synthetic polypeptide. So this is being done for those kind of polymers where cells do not adhere, because those polymers lack the sites through which that will target the integrins. Okay, so like the RGT peptides, etc., they specifically add onto that polymer, or chemically add, means by covalent bonding. These RGD peptides and other types of peptides are there, which are grafted onto the polymer chain, onto the polymer backbone. Okay, and then the synthetic polymer can adhere cells. Okay, cells can adhere to the synthetic polymer. If the grafting is not done, then the cell won't adhere on. So this is one way of modifying the surface property. Another way is you whatever functional group that you have added onto your surface, onto your polymer, you can, so these green structures, these are some functional group, and those are connected with your surface with some long linker molecules, say carbon hydrogen bonds, etc. So you can modify the length of that linker. Okay, so in doing so, what will happen? Uh, If the longer linker if you use, then it will give more resistance to compression, right? And then your proteins in the solution will adhere less. And but if opposite is true, means if you use smaller linker molecules and you then the resistance to compression will be lesser, there will be higher chance of protein adhesion. So this is another way of modifying a surface property. The third way is you can create means you can um, add such groups onto the surface of the polymer or any biomaterial which which characteristics is changing in presence of some stimulus, like for example electric field. So this particular figure, so they have added one functional group, okay, and it towards the end it has some electronegative uh, group, and in presence of electric field, uh, this electronegative group, it basically changes this morphology. Okay, and due to the change, it will say before the in absence of electric field, it may not respond to any cell adhesion, but after the electric field is present, then some functional groups through which uh, the integrins receptors can bind, those get exposed and the cells can adhere. So in this way, you can control the surface property by using some kind of stimulant. Okay. Now you can also do amazing one more way. You can also use the cells. Okay, you can also use the cells to change your surface, biomaterial surface on demand. So, on your biomaterial, say you add some functional, well, some chemical group, some chemical molecule, okay, onto the surface of your biomaterial. But it is some inactive form. Inactive form, it is attached, and you are genetically modifying your cell to express one kind of an enzyme, and this enzyme is basically a transmembrane enzyme, means it is expressed on the surface of your cell. So you're doing genetic engineering onto your cell. So once this enzyme gets expressed and it is present on the cell membrane, then when the cells come in contact with the surface, this enzyme will act on the inactive chemical compound that you initially grafted onto your polymer surface or biomaterial surface. Then it will create a product. Okay? And this product will be like changing the characteristic of your particular biomaterial. Okay? So this is another way. Now, I think fifth way is you can use some carbohydrates. So this example is for specific for liver cell growth, where people have crafted some carbohydrate molecule onto the biomaterial surface, which promotes the adhesion or adsorption of a protein called lectin. So one lectin gets adhered, the liver cells can adhere properly on it and will grow nicely. So another way is you can use a cyclic form of RGD peptides rather than the linear RGD peptides to onto your synthetic scaffold, which does not have any cell binding sites. So people have observed the cyclic form is a better cell adhesion promoter than linear RGD. So you can play with the structure of the RGD for uh, triggering better cell adhesion. Okay. So any queries any one of you have regarding anything that we have discussed so far? No? Okay. So we'll move on with the next topic, that is characterizing your surface. So uh, whatever changes that we are adding, say we're changing the surface topology, or we are changing the groups, functional groups onto the surface, we have to do some experiments to confirm that those changes have happened properly. Okay, that is characterizations we need to do using some instruments, using some sophisticated techniques. So you can characterize a surface either its topology or its chemistry. Surface chemistry means what all functional groups are there. You can do it either using spectroscopy or using microscopy. And in microscopy also, there are a number of options available. There's optical microscope, like when you use light. There's confocal microscope, then two photon confocal microscope. Then you can use electron microscope. There's one mode called scanning electron microscope, where the electron beams falls only on the surface of the material, and you understand how, and then you obtain a picture. From that, you can understand how the surface looks like. Then there is scanning tunneling microscope. Then there's atomic force microscopy. And depending on how much resolution you want to go up to, means how less fine images you want to get out of your surface, you can choose among any of these techniques. Okay, where there is now microscopy will help you uh, understand how the surface looks like through images. You cannot quantify it very nicely. You cannot quantify the surface characteristics using microscope, but you can use the spectroscopic techniques for doing quantitative analysis. Like you want to like understand uh, how much of elements like the carbon, oxygen, nitrogen, etc. Those are present on your surface. Okay, so you want to quantify that, or you want to no, means spectroscopic techniques are mainly used for understanding the functional groups only. And we can actually go up to quantifying the elements that I just said. You can understand how many carbons, atoms, nitrogen, the distribution of carbon, nitrogen, oxygen, and other atoms, how they are distributed on the surface. You can do that using various spectroscopic techniques. So here also, people irradiated the surface of a sample, of the surface of a material, basically with photons like light, okay, or electrons, or they bombard it with beam of ions. And what they do, they measure. Then they measure how much ions or electrons or photons they are coming out from the surface. They measure that. Okay, so when you at very high speed and which is at a very high energy, when you irradiate, when the light beam is incident on the sample, it is forcing some atoms or electrons to come out of the surface, and that is being measured. What is being coming out? So in this kind of spectroscopic techniques, this is how it works. So a few example being the auger electron spectroscopy. Then there is photoelectron spectroscopy. So these techniques, they allow to understand what is surface composition, means what are functional groups etc. are present, by applying an incident beam at a angle, at different angles basically, so that you can understand, or you can cover as much surface as possible, so that you can understand what all atoms are there. You have to cover maximum area. Then only you can say with confidence that these kind of functional groups or atoms are present onto your surface. In auger electron spectroscopy, here they use a beam of electrons. Okay, and this beam of electron, what it does, they forces the inside one atom, they forces the inner atom present in the inner shell to uh, go up to a higher energy band. Okay, and an electron which is present at a higher energy band, it drops down to a lower energy band. So because of which a resultant uh, emission you get. You getting right? When the electron goes from higher to higher energy band, it requires energy. So the energy is coming from the incident electron beam, whereas when it drops, it gives some emission. So this emission is studied. Okay? Whereas in photoelectron spectroscopy, it is given other names like XPS, then ESCA. So here they also measure characterization helps you to get more accurate data than AES. Okay, so here they only study what kind of emission is coming, but here they also study the which electron is coming down and all. So those which from which band gap, which from which energy band. So that study, so that is also being measured. So that is why data that you get from XPS is much more accurate than AES alone for understanding the surface chemistry of your particular biomaterial. Now, we have now finished about the surface properties that one need to look into for a biomaterial. Now we will briefly see what kind of biomaterials are there. So when we started this topic, I already introduced to around three classes of big classes of biomaterials that are being used. That is metals, ceramics, and polymers. Okay, but here in today's lecture, I'll be categorizing the biomaterial based on the source from where they are derived. So the characterization that I gave previously, that is category that I gave previously, that is metal, polymers, and ceramic. It was based on what all primary bonds are present between the atoms that form these materials, whether it is metallic bonds or covalent bonds or ionic bonds, etc. Now here, depending on the source, we are categorizing the biomaterial. So natural materials means those materials which are derived from uh, natural sources like plants, animals, etc. And these materials, they are, even though not completely identical, but similar to the materials found in the biological environment. So, which is why the cells can easily recognize them and also process them through their own metabolic pathway. By process, I mean many of the natural biomaterials are biodegradable. They respond to the enzymes like mattress metalloprotease enzymes, and they are polymer chain. They are basically natural materials. Most of them are polymers in nature. So whatever bonds are there between the monomers, they can be easily broken down by these enzymes and turned into simpler units, which the body, which is usually not toxic to our body, and it can be easily flushed out. Okay, is either in form of some urine or feces, etc. Okay. Now, there are various types of natural materials that are available. You can again categorize them like proteins, like silk, collagen, elastin, fibrin. So these all examples have been used for tissue engineering application. Then there is polysaccharides. So there is chitin, there is GAG, alginate. So these are again. So alginate is a plant-based, like it is taken from seaweed. Chitin is taken from the crustaceous animals like the crabs or prawns. So from their shell, chitin is get. We get chitin. Then glycosaminoglycans. So this is again taken from animal tissue. Okay, but also people have made like tissue engineering scaffolds, or you can say even composites of or like of the material using DNA and RNA incorporated in them. Okay. so that they can also act as biomaterials. So this is a new area of research that maybe if any one of you find it interesting can look into. So what are advantages are there of using a natural material? First is uh, it is biologically active, means the our body can recognize it. Okay. And it can since it is most of this material like collagen, elastin, etc. GAG, so these are already present in our body. So usually our body do not recognize it as foreign. Okay, so it does not invoke very high inflammatory response, very intense inflammatory response is not invoked by this kind of materials. Also, they can be degraded by the naturally occurring enzymes in our body to simpler, easily removable chemical compounds. But it also has some disadvantages. So, first disadvantage being if you are using a protein, say like collagen from say rat tissue. So rat tissue, and you're using that collagen for human, then there can be some immunogenic response because uh, when a protein from a like rat and a protein from human, there will be minor differences in the structure of the collagen. Okay, and this difference is species specific. So because of which it can invoke immunogenic response in our body. Okay. Now, but also for making this kind of material, and when you purchase it or when you extract it, you will see that the quality of the material varies from one batch to another. So, when you use this material for your application, then the results might also vary. Okay. And if you want to manipulate this material, say add any functional group, etc., then the process is quite elaborate and complicated. All right. And also there are synthetic materials. Synthetic materials are those materials that you synthesize in a lab, like a chemist or material scientist help you synthesize it. Okay. Now, again, you can divide it into what all types of synthetic materials there can be, like the metals, ceramics, then there are polymers, and a composite of metal, ceramics, polymers. Okay. So, metals examples like the titanium and its alloy. Then there is also a use of stainless steel, etc. The magnesium alloys are also used. Under ceramics, you can use hydroxyapatite. So this is a calcium phosphate, uh, basically two groups, calcium, two atoms, calcium and phosphate, that are present at different concentration. It forms hydroxyapatite. All right. Then there are also polymers. Now, polymers can be synthetic, like the polyesters are there, like polylactic acid, polyglycolic acid, polycarbolactone. Also, the example that we talked about previously, the PEG, that is also a synthetic material. And you can make a composite of either metal polymer or polymer ceramic, etc., for your particular application. And it has some advantages. Their use has some advantages. So it is relatively easy to uh, design experiments for modifying the structure or the chemistry of these synthetic materials. Okay, means people do have expertise, and whatever changes that you do, they are reproducible in nature. And also, synthesis of this material is highly reproducible. You can pinpoint uh, onto everything. You can and you can figure out what went wrong while working with this kind of materials because the composition etc is fixed, entirely fixed. You know the polymer chain, how it looks like, what all groups are there. Everything is well defined for synthetic materials. That is why whatever results you obtain using these materials, they become highly reproducible. and they offer very good mechanical properties as well. Okay? But it has some disadvantages. First, very important disadvantage is if you are going to use synthetic material for biological application where you want your cells to grow and then ultimately form tissue, then it is not possible because these materials they do not have the required cell binding sites. Okay? So you have to graft say synthetic polypeptides like RGD etc or you can like do other modifications so that our body can recognize it, okay, and or the cells that you want to grow on it. So you have to modify it in order to get some in order to make it biologically active. And also, they do not contain any groups which respond to the naturally occurring enzymes, and which is why they are not they either cannot be degraded by the body or the degradation process is very slow. Okay, so this is a table which shows you the types of biomaterials that are being used for different applications. Okay, so metals like titanium, alloys, then cobalt, chromium, alloy, stainless steels. So these are all used for making knee replacement prosthetic, knee, for making total artificial knee basically, then artificial joints. So those people who are having osteoarthritis, etc., and their knee and knee joints or hip joints they are not working properly or they have corroded very badly. So for them, people are making this kind of um, implantable uh, structures using this type of materials. Then for dentists also use various kind of ceramics like alumina, zirconia to create structures, okay, for various dental applications. Then for heart valves, for making heart valves and pacemakers are made from various kind of polymers. See, like polyurethanes, polyesters are used. Then the lenses that not the contact lenses that usually people advertise that you can apply from the top, but the lens through the interocular lens means that is inside your eye. Okay, uh, that lens if it gets damaged somehow, so that can be replaced, and those are artificially made using polymers like PMMA, okay, then silicone, etc. Then vascular grafts like uh, a small section of a blood vessel that is completely damaged, or due to uh, atherosclerosis, some uh, lipid molecules they have clogged up the um, your blood vessel. So you can then remove it, okay, and in place you can add some vascular graft, and they are usually made from polymers like dacron, okay, polyurethane, etc. And other for making electrodes or for making biosensors also, various polymers are being used for drug delivery systems where you need a drug to be released at a very sustained level. You can use various kind of metals or ceramics. So these are some examples of applications where the synthetic materials like the metal ceramics, polymers, or their composites are being used. So that's it from my side in today's class. If you guys have any queries, then let me know. So, any queries? All right. So, no queries. So I will just then simply. Uh, so you can. Are you guys there in the meeting?
Yes, ma'am.
Yes, ma'am.
So, let me just take your attendance for today's class. I did you guys understand everything, or you guys didn't understand anything? That is why you didn't get any queries. So, which one was it? Understood everything, or didn't understand anything? Because if in either of this case only, there can be no queries. It's totally fine. Like we have come so far that you can be frank with me. But, um, so who all are present? Quickly respond with your names.
Ma'am Kirtan.
Okay, Ethan is there. Next, who is there in the
Vani.
meeting? Um,
Ma'am Harsh.
Harshita. Okay. So I have marked for Ketan, Vani, and Harshita. Anyone else is here? So,
Ma'am
so
Nidhi.
Nidhi. Okay. So only four of you is here. That's fine. But just very frankly, I'm asking: are you guys following what I've been talking about in the past 2 3 classes?
Yes, ma'am.
Okay, all right, that's good.